Good afternoon, everyone, and welcome to the University of Manitoba Alumni and Friends Virtual Learning for Life Fall 2020 series. My name is Tracy Bowman. I'm the Director of Alumni Relations and also a proud alumna and the moderator for today's session. Thank you for joining us and making this part of your day. We've been able to offer this program free of charge to our 148,000 alumni living in 140 countries worldwide, thanks to the very generous sponsorship of one of the UM's affinity partners, IA Financial Group. Many thanks to them. Delivering learning for life opportunities is a very important role for the University of Manitoba, and we're so very proud that we're able to feature so many of our leading researchers and professors in this way. Now, just a few housekeeping details before we move on to our speaker for today. And that is, as usual, you're watching this session in YouTube. We will be sending you, we will be recording this and we'll be sending this link to you afterwards so you can view it again if you'd like. You were sent the link to a platform called Slido. That is www.sli.do with the password VL18. And the reason we sent that to you is that is how you're able to ask questions of our speaker today. I will be monitoring that platform and I will be reading the questions on your behalf. Of course, please ask as many questions as you like. Please send follow-up questions. If perhaps I've misrepresented your question, ask it again, and we will try to get through as many as we can in the time that we have. So now I'd like to introduce our speaker for today, Dr. Keith Fowke, who will be presenting on the topic of research on preventing infectious diseases, studies from HIV and COVID-19. Now, let me tell you a little bit about Dr. Fowke. He is a two-time UM alumnus, and he's also the head of the Department of Medical Microbiology and Infectious Diseases at the Rady Faculty of Health Sciences. He's also a visiting lecturer in the Department of Medical Microbiology at the University of Nairobi, and I'm sure he'll be sharing about those experiences with us today. Dr. Fowke has established an active research laboratory that is currently focused on a number of different research areas. And I've paraphrased a bit of this. And so Dr. Fowke, if I've gotten anything wrong, please let me know and you can share with our guest today. Those research areas include determining immunological phenotypes in individuals who are resistant to HIV infection, how to induce this low immune activation phenotype by using anti-inflammatory drugs to reduce the number of HIV target cells, and also how HIV dysregulates the immune response and identifying suitable targets for intervention. And so for these projects, the laboratory has received numerous local, national, and international funding, including 2 CIHR grants. So with that, over to you, Dr. Fowke.
Great. Thank you very much, Tracy. And thanks for the invitation to speak today. And also thanks to Dustin for reaching out and coordinating with me at the beginning and all the technical help in the background. Thanks very much for taking time out of your schedules to attend this talk, and really appreciate your interest in what's continuing to go on at the University of Manitoba. And we're building on the legacy of many of our alumni. So thank you for your past contributions as well. What I'll do today is just touch briefly on 2 topics, HIV and COVID-19. And I'll do it from a very narrow lens of the research that's involved in my own lab. And that'll be the focus of today. But to start with, just have to wake my computer up. There we go. I'm also the head of the Department of Medical Microbiology and Infectious Diseases, which is based on the Bannatyne campus, which is adjacent to the Health Sciences Center downtown. And we're a small department. We have 9 full-time basic science faculty members, but we're highly collaborative. We have 109 other members, and we have members that are clinicians, people from other departments, and also from different aspects of public health research. We do basic science, but we also partner with people doing clinical infectious diseases as well as public health. And some of our main public health partners include the National Microbiology Lab and the Canadian Food Inspection Agency, which are part of the big White Virology building on Arlington. And also we're partners with Chatham Provincial Laboratory. So I think across Canada, we're unique in that we're not only basic scientists, but we're also linked with clinical and public health very tightly. Our department has 3 areas of research expertise. The first is antimicrobial resistance, so that's superbugs, how bugs are becoming resistant to the antibiotics that we use. The next area is viral pathogenesis, and we do research in Ebola, Zika, and as you'll see a little bit today, COVID. And then also our department has had a historical strength in HIV research based on the work of Dr. Alan Ronald and Frank Plummer, who pioneered that work in our department. And our department brings in a fair amount of research dollars each year. We have a number of department members who I'll just touch on briefly. Jason Kinderchuk is he's an Ebola researcher, but very much involved in SARS COVID discussions. And so he's always on the news and doing a great job of providing good scientific information to the general public. Denise Bay is an antimicrobial resistance researcher. Dr. Yao will be highlighted. I'll highlight some of his work later. He does HIV work, but he's also working on a SARS vaccine. And there's a number of other researchers. Zuma Rueda is a Colombian clinician and epidemiologist who's coming to our department once she's able to get on an airplane and fly north. So we look forward to her joining us. We have collaborators and members of our department in Kenya. Both Joshua Kamani and Larry Gelman are based in Kenya. We have lots of other members who are making wide contributions in all of those areas. So you should be, as an alumnus, you should be proud of all of their work. I know I certainly am. I just also like to highlight one thing that we're looking forward to, and it's with sort of mixed emotions that we have to have this event. Dr. Frank Plummer was a core member of our department and the university for many years, and he passed away early in 2020 while he was in Kenya. And so on December 1st, which is World AIDS Day, we're having a scientific symposium to honor him, but also to highlight the work that goes on that he laid the foundation for. So a lot of his past trainees will be presenting about the influence that Frank Plummer had on them, but also the work that they're currently going on. So if you're available, you'd be more than welcome to attend that. And the information is available, and you can email me anytime and I can provide details. So our lab does a number of different research projects, and I'd like to not get into the details of several of them, but just present to you some of the faces that are working on some of these projects. These are current University of Manitoba staff and students and future alumnus. So I thought I would take a moment just to highlight some of that. Some of our work in our lab is working on a cure for HIV and therapies for HIV. And we have Colin, Allison, and Shifa working on those projects. And Colin and Allison are both Vanier Scholars, which is the top scholarship that a graduate student in Canada can receive. And Shifa has a scholarship from Research Manitoba, so the Manitoba taxpayers are helping to cover that. So their projects are working on HIV cure. Another aspect that we're doing is in partnership with Sunshine House, which is an organization in downtown Winnipeg that works with street-involved people. We're trying to understand the effect of breathing solvents has on the immune system. And we have Monica Coacit, who has a Canadian Institutes for Health Research scholarship. And Jillian McGyver has worked on that. Jillian is now in med school. So this is a project about local partnerships, looking at some of the problems that we have here in Winnipeg and that are global in nature also, and how when people use solvents, what it can do to their immune system. We're also working with a Canadian team to look at an HIV vaccine. The leader is Gary Cobinger, who used to be here at the Public Health Agency lab at the National Microbiology Lab, but now is at the University of Laval. And with Dr. Yao and Dr. Ball and myself, we're collaborating with them on assessing HIV vaccines in a non-human primate model. And Monica and Natasha and Paula, a summer student, have been working on that project. I've done work in Nairobi, Kenya for my whole career, really, since 1990. And a lot of that work involves work built upon that of Frank Plummer, where we're working with sex workers in Nairobi. And we're trying to understand how HIV affects their immune system and how they're able to fight off HIV infection. And so Genevieve, Julie, and Ken, who's a PhD student in Nairobi, are all working on some aspects of trying to understand how HIV and sex work are related. We also have another team that's working, trying to understand how different hormones that are used to prevent pregnancy may impact HIV risk. And Julie Lajoie is a research associate in my lab. She really leads this aspect of the lab's work. And I'll also highlight Toby Lee, who is a master's student just transitioning to the PhD, and he also has a Canadian Institutes for Health Research scholarship. What I'll focus on today is two projects, one on COVID, and my lab has developed some systems to detect the antibodies of influenza virus, and we're going to be adapting that to detect the COVID virus also. And my technician, Steve Wayne, has done all of that work. And then the second part of the talk today will be focusing on a brand new approach to try and prevent HIV infection using aspirin. And this is the team, Ken and Lucy, our University of Nairobi students working on the project as well. So that's the team. And you can see that a lot of the students have been supported by taxpayer dollars through the Canadian Institutes for Health Research or through Research Manitoba. So we thank the taxpayer for supporting research. COVID-19. So everyone has heard lots about COVID-19. And I'm not going to spend a lot of time going into much of the details. There are experts at the University of Manitoba who are public health experts who know more about those details than I do. But I think we're all familiar with the first spike that we had in Canada, and now we see this increase in cases. And this data was as of yesterday. So the case rates are, daily case rates are increasing all the time. Just to remind people that the term COVID-19 refers to the disease that a person gets. The virus that causes that disease is the SARS coronavirus 2, type 2. And this is what the virus looks like, or at least this is my rendition or interpretation of what the virus looks like on Halloween. I think maybe a little less festive version of that is this diagram where it shows the virus, and then these red bits sticking out are what's called the spike protein, and that's what the virus uses to attach to a cell to infect it. And as we'll discuss, HIV or vaccines against SARS coronavirus often incorporate part of this spike protein. So globally, there's 22 million people have been infected by the virus, and 1.3 million deaths as of yesterday. In Canada, 306,000 cases and 11 million deaths. And in Manitoba here, we have, again, this was as of yesterday, 11,608 cases and 179 deaths. So it's something that's affected everyone, every aspect of all of our lives. And I would say that all of those researchers that I introduced earlier almost all of them have moved in some way to be doing COVID-19 and SARS coronavirus research. Dr. Jason Kinderchuk is actually relocated for 1 year to Saskatoon so that he can access some high containment labs and do some work there. We're trying to build new labs at the University of Manitoba here to allow him to continue doing some of his high containment work. And so we're very pleased that we're going to be able to renovate a lab and so that he can work with the SARS coronavirus directly here, and other people like myself also. So when we think of COVID research, I think of really 3 different areas. There's many more. Of course, there's all different kinds of research, including social sciences, which is really important, how people's affecting their mental health and different things. But from a laboratory medical microbiologist point of view, I'm really thinking of it as detecting the virus, preventing the infection, and then treating people that are infected. And if we think about prevention, what first comes to mind are vaccines. And here's a cartoon of an Ebola virus. So here's the virus. Not quite as nice as my pumpkin in my view, but still it's nice. So here we have the red spikes that are on the outside of the virus. And then inside the virus is the genetic material of the virus. And you can see this little red bit of genetic material here. The gene here codes for this particular virus. So the whole idea with a vaccine is that we produce, the immune system recognizes parts of this virus in advance to actually being exposed to the live virus itself. And the immune system generates, it fights the infection by producing antibodies. That's these proteins here. And these antibodies will bind to the virus and then eliminate it from the system. And that's typically of what we think of when we think of vaccines. However, if you also, some vaccines target the white blood cells of the body. So the white blood cells are, one type of them are lymphocytes. And if you have an infection and there's all kinds of pus in that infection, all that pus is really white blood cells coming to the site of infection to fight it off. And vaccines can be targeted so that the white blood cells of the immune system can fight off the virus as well. In general, very simplistic terms, there's 3 different types of vaccines. One is something called mRNA, and that's where they take some genetic material of the virus, just a small bit, and they inject that into a person, and I'll talk about that in a little bit. Another type of vaccine is a subunit vaccine, and that's where they actually produce part of the protein of the virus, the spike protein, and they inject that into a person to cause it to the body to recognize it and mount an immune response to it. And then the other type of vaccine is a vector, and this is a safe virus that they take out the little chunk of the SARS coronavirus that encodes the COVID, and they transfer that into a second safe vaccine, and then they use that, a second safe virus, and then that virus is used to deliver that piece of SARS coronavirus material into the cells and generate a vaccine. So all of these 3 different kinds of vaccine types are being tested today, and I'll talk about some mRNA ones, and then also a little bit about these other 2 from some research that's happening in our department. So in terms of vaccines, vaccines have to go through a complicated testing process. Phase 1 is it safe? Phase 2 is it stimulating the immune system? Phase 3 is, does it actually protect from the infection? Limited means that it's permitted to be used in very special circumstances. And so this are the numbers of vaccines, and then also, even prior to phase 1, before it goes into a human. That's called preclinical. And there's hundreds of vaccines, including one that I'll describe today, that are in preclinical, where they're being injected into animals to see if they are able to generate immune response and if there's any harm or anything. But to date, there's none that have been approved. However, I'm sure you're familiar in the news that there's been some recent press reports about some positive news in coronavirus vaccines. The first one is a Pfizer vaccine. And this is an RNA vaccine, so that's part of the one where it delivers the genes of the virus directly into the person. It's delivered through 2 doses, and importantly, it requires very cold storage in order for the product to remain stable. The RNA is it can be easily degraded, and if temperatures get too warm, then the vaccine can be degraded. So that's why the temperatures have to be really cold. There hasn't been a scientific publication, peer-reviewed publication out yet, but the press release from the company indicated that they had over 43,000 participants in their vaccine trial. And they said that there was diversity, and I assume that means in diversity in terms of age, gender, and ethnicity. But we'll have to see the details. And they claim greater than 90% effectiveness to date. So that's quite positive. In order for a vaccine to really have the kind of effect we want it to have, it really should have at least an 80% effectiveness. So this 90% of effectiveness is encouraging. So what that means is that 10% of the time, people that are given the vaccine, it doesn't actually work, and they still end up getting the infection. And that's what we saw with the second vaccine that's been recently announced from Moderna. Moderna also is an RNA vaccine, but the way they produce it, it's stable at 4 degrees, which is good. They still are going to ship it at a very cold temperature, but it seems to be a little bit more stable. They've enrolled 30,000 people in their study, and again, they've only released a press release, so no scientifically peer-reviewed data. But in those 30,000 people, they saw that 95 were infected, and some people got the placebo, and some people got the vaccine, and 90 people who received the placebo ended up getting COVID-19 infection, whereas only 5 who received the vaccine went on to get the COVID vaccine, get the COVID infection. So again, a vaccine isn't a guarantee that someone's not going to become infected. What we don't know are the characteristics of these 5 individuals who got the vaccine and didn't get infected, what their age was, or anything like that. But overall, it indicates, again, an over 94% effectiveness. So that's quite positive. There'll be challenges in terms of rolling out the vaccine, and some issues to keep in mind is these vaccines, even if they are proven to be effective, the production has to be ramped up. And I think that's one good thing about RNA vaccines, is that their production is going to be a little bit faster than maybe some of the other vaccines. The number of injections, 1 vaccine versus 2. Sometimes you need to get a second shot, which is like a boost. And it's going to be important to determine that. Heat stability of the vaccine, delivering vaccines to remote areas all around the world. It would be very challenging to do that if the material had to be kept really cold. So that's going to be a challenge. We don't know how long the immunity is going to last. Will the immunity last 6 months, a year, or will it be lifetime? I doubt it'll be lifetime, but hopefully it'll last, and we still don't know about that. We don't know the age that these vaccines work best at. We know that people over 60, 65 years of age are more likely to get severe symptoms if they're infected with COVID. but will the vaccine protect them? Will the vaccine work in people of all ages? We don't know that. And then finally, prioritization, who is going to get the vaccine first? There's lots of research that needs to be done in all of these areas. So there's still a lot of work to do, but to be honest, I didn't think that we'd be getting vaccines in our arms until the fall of 2021, but now I'm revising that based on these positive results, and hoping that in the spring of 2021 we'll be receiving vaccines, at least the people that are of highest priority, being healthcare workers and those at greatest risk of severe disease. So things are moving along. In terms of, I'll just give you a snapshot of some of the research that's going on. Again, a very narrow window here. This isn't all research that's occurring at the University of Manitoba. This is just some of the things that my lab has been involved in. And again, in the themes of detection, prevention, and treatment. So in terms of detection, I'm doing a project with collaborators at the University of Waterloo, Carolyn Wren and Emmanuel Ho. And this is, again, funded by the taxpayers of the Canadian government. So thanks for that. And this is a grant that they received to, I'll just read it out first, development of a microwave-enabled bio-nano-fluidic device for point-of-care diagnostics of COVID-19. So that's quite a mouthful. So what does that mean? So basically, this is trying to detect the virus using a microwave technology and using very small levels of fluids, so microfluidics, so very a device that you can use just a small amount of fluid. And point of care is that you could go to a doctor's office and the test could be run right there. So it's a test that is supposed to be easy to run, doesn't require a complicated lab to run it, and it's uses very small amounts of fluid and uses the microwave technology. Full disclosure, I know nothing about microwave technology. I know things about viruses and the immune system, and so that's where we collaborate with great people like Carolyn and Emmanuel who do know that. So, and briefly, someone is tested, they would get a swab up the nose that would go into some media, and then that media would be put onto a slide here. This is about the size of a microscope slide, so maybe 6 or 7 centimeters long by a couple centimeters wide. And then on this slide, there's a detector that detects using microwave resonance to detect it, and the signal is sent to an analyzer, and then the data comes out. And what this detector does is in the detector they have small pieces of gold, and those gold have antibodies against the SARS coronavirus. So again, these antibodies are very specific. They'll only bind to the SARS coronavirus. And when a sample that has viruses run over it, it binds and it changes the resonance of the product. Here's a case where the lab in Waterloo, they took just some saline and they dropped it onto this resonance chamber. And then in a second case, they took some SARS coronavirus proteins, not the live virus, but just some proteins, and they put it onto a second sample. And what you can see here is in blue is the sort of the placebo or just the water effect. And even when you increase the amount of virus or increase the sample amount, there's no change. But when you use SARS coronavirus, you can see that they're able to detect it. So the goal is really to increase the sensitivity of this assay so that the virus could be detected. So this is one of a whole host of projects that are looking at trying to detect the virus very early on. In terms of prevention research, one of my colleagues at the University of Manitoba, Dr. Xiaojian Yao, as well as Darwin Kabasa and Sam Kung, we're doing a CHR-funded project on the development of a vaccine. So this is a novel dendritic cell targeting vaccine that targets the spike protein of the SARS coronavirus. To walk you through this title, basically, the dendritic cell is a main cell of the immune system, and so this vaccine is specifically trying to deliver the spike protein of the coronavirus to this key immune cell with the goal of having it to increase the ability of the immune system to recognize this. So the Dr. Yao's lab is approaching this in a couple of different ways. In the laboratory, they grow in some human cell lines. They grow just the SARS coronavirus spike protein, so just that little bit that sticks out of the protein. They grow it up in the laboratory, purify it, and then inject it into mice. And when they do that, they can see that the mice generate an immune response against the spike protein. So this is an example of a subunit vaccine where a little bit of the viral protein is produced and then injected into the host. The next thing that they're working on is a vector vaccine. And this is using a very safe virus called vesicular stomatitis virus. And VSV virus has been used for many different vaccines. And actually our colleagues at the National Microbiology Lab pioneered the Ebola vaccine with the help of the University of Manitoba, and students at the University of Manitoba did some of that work. So this is basically a very similar vaccine where they take a small bit of one of the Ebola virus proteins, and this bit causes the dendritic cells of the immune system to recognize this protein and basically feed it into the immune system. And what Dr. Yao's lab is doing is taking the COVID spike proteins and putting it into the system, so that what is produced is a VSV virus that has little bits of the spike protein of COVID, and it has little bits of the Ebola virus, none of which would cause the full infection or any kind of infection. But what they do is they target the immune system very effectively. And so it's a way to specifically deliver this vaccine to very important cells of the immune system. And then downstream, my role will be to help characterize the immune system, the immune response once it's injected into mice. And then if positive results occur, then maybe it could go on to be used in humans and tested in humans. But the fact that this is the same backbone that was used for the Ebola vaccine, I think is quite promising that it's safe and it's effective, and maybe we can adapt it to look at COVID. Some other research that we're involved in is in terms of treatment. So you may have heard early on in the epidemic, a certain president of a certain country talking about hydroxychloroquine and how everyone should rush out and use it. Hydroxychloroquine is well known to reduce the immune response, and it's actually used to treat autoimmunity. So people that have rheumatoid arthritis will often take hydroxychloroquine to dampen the immune system so that their autoimmunity doesn't flare up. However, there's been studies now that looked at once someone is infected with COVID, does hydroxychloroquine help? And the results have come out that contrary to hopes, no, hydroxychloroquine doesn't help once someone is already infected. However, can it be used to prevent infections in the first place? So that would be a prophylactic or preventative approach. And we don't know. There's trials that are ongoing that's looking at that. And actually, as part of our HIV studies a number of years ago, long before COVID came along, we actually did a study in normal human donors to see the effects of hydroxychloroquine. And so we have some data for that. So its role in normal donors is not really known or understood. And so our lab looked at people who were, we studied them for a month before they received hydroxychloroquine, and then we followed them for 6 weeks afterwards. And then we looked at many different aspects of the immune system. And one thing we wanted to understand is, does it interfere with antibodies that someone might have produced? So H1N1 is the influenza virus. H3N2 is another kind of influenza virus. And what we did here was we looked at baseline. So this is before people received the hydroxychloroquine. And then we looked at them 2 weeks after they've been on hydroxychloroquine. And again, 6 weeks after they were on hydroxychloroquine. Compared to the baseline before they were on it. And here we saw that for H1N1 antibodies, as well as H3N2 antibodies, taking hydroxychloroquine didn't have any effect on pre-existing antibodies. However, when we looked at total level of antibodies, and the class of antibodies that we looked at are called IgG. It's a very stable, long-lasting class of antibodies. What we saw is that after 2 weeks, the amount of total antibodies in someone's blood actually decreased, and it stayed down for up to 6 weeks. So this suggested that hydroxychloroquine may have been preventing brand new antibodies from forming and keeping them down, which is, in a way, it's a good thing because that shows the immune response is being limited, but also would be a concern if someone was taking hydroxychloroquine and also wanting to take a vaccine, then that our data here would suggest that the hydroxychloroquine may interfere with their ability to produce a new antibody response. So the influenza-specific antibodies that were already in a person's body, they weren't infected, but the total amount of antibodies in someone's body dropped, and this is likely because they weren't able to produce as many new antibodies. We also looked at the white blood cells of the immune system, and on within the first 2 weeks, we saw a decrease in immune cell activity, but importantly, a lot of the activity returned back to normal by 6 weeks. So we do see hydroxychloroquine having some short-term effects on the immune system, but over time, those seem to wane. So I think where these studies are important is that when people are deciding whether they should be taking a drug long-term to prevent an infection, you need to understand what's the long-term and the short-term effects and benefits of it. And so I think our study that we did 4 years ago can help inform people about whether hydroxychloroquine would be helpful in preventing infections. And we're just in the process of finalizing the paper that we'll submit for publication. So that's all I have to say about COVID. I'd be happy to take some questions afterwards. And now I'll turn my attention to HIV, which is where I've been studying for the last 30 years or so, and 20 as an investigator here at the University of Manitoba. So some statistics, there's 38 million people living worldwide with HIV. And in the last year, there's 1.7 million people were newly infected. And that works out to be 4,500 people infected every day throughout the world. AIDS deaths are about 700,000. So we saw in the statistics for SARS coronavirus much higher levels of infection and death. But that, I'll get back to that, wasn't always the case for HIV. So I think most people are familiar that most HIV cases are found in Sub-Saharan Africa. About 2/3 of all cases in the world are found in Sub-Saharan Africa, and it's found all over the world. If we look at, we look back in time, 1990, 1995, 2000, and this is the number of AIDS deaths per year. So this is 1 million, 2 million, 3 million. So in 1990, the virus was just beginning to spread. By 1995, there was 1 million people were dying of HIV every year. And then by the year 2000, almost 2 million people were dying. So this is when I got involved, and this is a very concerning situation that we didn't seem to be able to stop people from dying. But then the HIV field developed medications that could stop the replication of the virus. It doesn't cure people of HIV, but it stops the virus from replicating. And we saw that people weren't dying as much. There were not as many deaths. So that's great news. However, what we haven't done as well on is decreasing the number of people that are getting infected. So in 2000, there was about 2.5 million people that were infected annually. And as of this year, it was 1.7 million. This is the goal that the UNAIDS had set for 2020 of 500,000 people infected every year. And we're not anywhere near that goal. So we still need to do a lot in terms of HIV prevention. In Manitoba, there's about 2,000 people living with HIV, and about 115 new infections occurred in 2018, which is the last year that we have statistics for. A little bit about the epidemic in Manitoba. There's a disproportionate amount of number of Indigenous individuals who were infected with HIV. In 2016, it was 38%, and in 2018, it rose to 51%. So despite Indigenous community only representing about 14–15% of the population in Manitoba, they represent half of new cases of HIV. And that gets to, HIV is found in very high levels in lots of different communities, and especially it's associated with poverty and low socioeconomic conditions. So there's many social factors that go into this, but the epidemic in Manitoba is very distinct in that we have so many Indigenous people that are getting infected. So obviously we want to prevent infections, and there's a number of different ways that we can prevent infections, such as giving people antiretroviral therapy, treating their sexually transmitted infections, abstinence, be faithful, using a condom, and male circumcision, which is work done by Stephen Moses and Frank Plummer at Manitoba. There's all ways that people can prevent HIV infections. We're still looking to develop an HIV vaccine and microbicides. Microbicides are products that women can add to the genital tract that would kill the virus once it's introduced. And we're still looking for good products. So this is the current tool of prevention approaches. And for some people, these approaches aren't sufficient and we need more options. So at this very basic level, if we want to prevent an HIV infection, this is what we have to prevent. This is a viable virus and it's coming into contact with a susceptible cell. And only certain cells of the body can be infected by HIV. And we have identified markers on the surface of them, we call them CD4 and CCR5, that allow the virus to get entry into the cell. So if you want to prevent HIV infection, you can focus on keeping the virus away, using condoms, treating with drugs, microbicides, and also male circumcision. This is all focused on the virus, but there's very few approaches that focus on this half of the equation, which is the susceptible cell. And that's what the project that I'll describe today is looking to do. So this is a diagram a very rough diagram. If you think of the female genital tract, this would be the cells of the genital tract. This would be the vaginal vault, and this would be the tissue underneath. And what many research has shown is that if there's lots of cells of the immune system here in the, just under the tissues of the genital tract, these cells, these CD4 cells become targets for HIV infection. These cells get here because of inflammation. So if someone has a genital infection or maybe a bacterial infection, that can cause the cells of the immune system to come here. And if these cells are turned on and activated, HIV can easily infect them. The work that we've done in Nairobi has shown that there's some sex workers who are highly exposed to HIV, but they don't get infected. And their immune system is characterized by having mainly quiet immune cells that aren't susceptible to HIV infection. So it's quite clear that inflammation, the process that takes immune cells from the blood and brings them to the tissue, that process leads to more HIV infection. So the way I think of it is that a cell that's moving from the blood into the tissue is an active cell. That cell has been turned on, and I think of it like a factory. And if a factory has a workforce, supplies, energy, shipping products, shipping protocols, production protocols, all of that is ready, and the virus is able to come into this active factory, give new production orders, and then this factory is able to be infected by HIV and produce new viruses. However, a resting immune cell, a cell that's not active, and it's sort of just sitting in the blood or in the lymph nodes of the body and is not going into the tissues, that factory, that cell, doesn't have a workforce, energy supplies. And so HIV finds it very hard to infect this cell. So when we think of inflammation, what's happening is that an active cell is moving from the blood into the tissue, and that's where it's being infected. So here we have a situation where we have inflammation, which causes HIV risk, and we want to bring the inflammation down to a state that we call immune quiescent, which just means quiet immune system. But we don't want to move it down to the point where the immune system doesn't work any longer, which is immune suppression. So we're trying to find ways that we can bring down the level of inflammation until we reach this sort of resting, quiet immune state, which is similar to the sex workers in Nairobi who are exposed to HIV, but they're not infected. So the whole idea of this study is, how do we reduce inflammation in the genital tract, and can we use some anti-inflammatory drugs to help us do that? We're looking for a drug that has to be safe, it has to be affordable, accessible, it has to be available throughout the world, and people have to be willing to take it. It needs to be acceptable. And the drug that we chose to study is aspirin, acetylsalicylic acid, or ASA, which we all know treats fever and headaches, but can also be used for prevention of cardiovascular disease, and used very safely at a low dose of 81 milligrams per day. And in consultation with the community of women in Nairobi, they said that this was something that they were familiar with and they would be willing to take it, whereas some of the anti-HIV drugs are stigmatizing and they're not willing to take those. So we did a study in Kenya, in 2 different clinics in Kenya, and we followed 37 women, and we followed them for 1 month when they weren't taking anything, and we took their blood and studied their immune system of the genital tract also, and then we gave them aspirin, low dose, 1 pill per day for 6 weeks. And here we followed them for 6 weeks on the drug. So each woman is compared back to themselves from this sample point to this sample point. And there were no drug-associated adverse events associated with this small study. We know that the women were taking the aspirin because it got into their blood. This is the blood level of aspirin. But we didn't know if it would actually get to the genital tract, which is really where we wanted it to. And we did see that levels of aspirin did show up in the genital tract, and this is the first time that was shown. So the women were taking the drug, and it did show up at the genital tract. We know that the drug was doing something. This is thromboxane, which is a protein that is downregulated in the presence of the drug. So at the baseline level, before women took anything, their drug levels were, their thromboxane levels were this level. And then they took aspirin and it dropped down. So thromboxane levels dropped due to the aspirin. And then visit 4 is when they stopped taking the drug 2 weeks later at the end of the study, and these levels bounced back up again. So this shows that the aspirin was being taken and doing something in their blood. In particular, we were interested in studying the number of HIV target cells in their genital tract, and these are the cells that we're trying to detect in their genital tract. And the bottom line is that we saw, depending on the type of immune cell we were looking at, we saw a 35% decrease in the amount of HIV target cells in their genital tract in women that took aspirin, or 28% if we look at a different kind of HIV target cell. So that's quite encouraging. Also, if we talk about the factories, an activated cell is a sort of one of those active factories. The immune cells were less active after taking aspirin. So markers on the immune system like HLA-DR or CD69 decreased. So not only were there fewer cells, but the cells that were there were not as likely to be infected by HIV. And here is the number of cells that we find in the genital tract. So this is 50%, 40%, 30%. And here is the amount of aspirin that's found in the genital tract. So if someone has a lot of aspirin in their blood, in their genital tract, they have very low levels of HIV target cells in their blood. Whereas if someone didn't have as much didn't have as much aspirin reaching the genital tract, they were more likely to have higher levels of HIV target cells in the genital tract. So this study showed that that by taking low-dose aspirin, we were able to reduce the number of HIV target cells in the genital tract, and that the level of aspirin correlated with the reduction in the number of cells. So our lab is continuing to study this. We're going to do another study in 2021 looking at high-risk female sex workers, and we're going to compare a couple of different doses of aspirin and look at it over 6 months, and also see if there's any additional effects when we combine it with other HIV prevention approaches. My lab continues to try and determine the mechanism, exactly how is aspirin affecting the immune cells. We have collaborators that are looking at tissue models. So they take a piece of tissue and infect it. And here, if they use a large amount of aspirin, they see a large amount of inhibition. So this is 90% of the virus is inhibited. And then as you use less amount of aspirin, there's less inhibition of the virus. So it shows that the more the more aspirin you use, the more you can block HIV replication. Other collaborators at the University of Manitoba are studying humanized mouse. So this is a mouse that has a human immune system, and we're going to treat that with aspirin and then challenge with HIV. And Emmanuel Ho from University of Waterloo is, rather than taking aspirin orally, looking at delivering it through an intravaginal matrix, which should decrease any systemic effects and have the drug only available locally. So the advantage of using this aspirin approach is that people are familiar with aspirin. It's not stigmatizing. It's cheap and affordable. It's already in every small kiosk throughout the world. You don't have to develop a -70 distribution network for it. It's already there. And it can be used with other approaches. Important considerations is that we're trying to reduce inflammation, but we don't want to get to the point where we're causing immune suppression. And I don't think that's, we're at that. I don't think that's happening. And it should also not be a standalone prevention. Someone should choose many different options from the HIV prevention toolbox. And aspirin doesn't make sense for everyone. Someone highly exposed, like a sex worker, maybe taking aspirin every day makes sense. But for someone that's at very low risk, then it doesn't make sense to take aspirin every day to prevent HIV. So this is the toolbox that we currently have. And what we're hoping is that this new concept of targeting the immune cell rather than the virus and reducing inflammation is a new way of preventing HIV infection and a new tool that we can use in the toolbox. So in conclusion, we're trying to use blocking inflammation as a new HIV prevention approach, and we're doing COVID-19 research to try and detect it, prevent it, and treat it. And these are members of my lab who I introduced at the beginning. I'd like to thank the participants in our studies in Nairobi. And of course, I'd like to thank all of the members of my lab and collaborators, as well as funders, and of course the University of Manitoba for being the best place in the world to do HIV basic science research. Thank you.
Thank you so much for that, Dr. Fowke. That was really fascinating. So one question earlier during more of your COVID discussion. So I encourage people again, please ask away. Please do post your questions on Slido.com. Password is VL18. The one question that came through, as I said early on, is what is the rate of effectiveness for the yearly influenza vaccine? And so really what they're wanting to know is a comparison to COVID vaccine. Yeah,
I mean, it's a good point. Influenza, as I showed, there was a couple different versions of influenza, and we actually, in that study, we actually test for, I think it was 12 different versions of influenza. There's hundreds of different versions of influenza, and depending on if they guessed right when they were producing the vaccine 6 months before the flu season, if they guessed right, then the effectiveness of influenza can be, if it's a good match, it can be, 60, 80%. But sometimes the match isn't good, and they didn't guess right, and then we're down into the 50%. So that, if you take it, then so for example, if it's 50% effectiveness, you still could be protected from getting the infection, but it's not guaranteed. And really, for a whole population level, you want that effectiveness to be increased. Again, I think what COVID has really taught us is that the washing your hands, not shaking other people's hands, wearing a mask, I think that's going to go a long way to decreasing influenza season this year. I hope so anyway. But you're right, the influenza vaccine ranges, I would say, probably from 50 to 80% effective. Okay, that's good to know. Okay, a few more other questions have come through. Okay, does the vaccine kept at -70 stay liquid or need to be thawed? And does it freeze flesh, at the injection site? Yeah, no, they would take the vaccine would have to be transported and stored at cold temperature, and it would be in a frozen state. Then they would quickly thaw it, and it would be liquid, so it would be obviously above 0 degrees Celsius at the time that it's injected. Whether or not there is any, whether they warm it up to 37 degrees or if they keep it cold when they're injecting it, I'm not sure, but for sure it will be in a liquid state when they inject it. And sometimes there's some inflammation around the injection site. I got my flu vaccine yesterday, and as I'm touching my shoulder here, I can feel it's a little bit sensitive. Again, that's cells of the immune system rushing to that area because you just stuck a needle in your arm. And whether this vaccine has inflammation, we'll still have to see, but it'll be injected at a liquid format.
And when is likely, I mean, we've seen a lot on vaccines and a variety of different companies that have been noted in the media, particularly the last week. I mean, what are we looking at realistically in terms of timing, in terms of availability, and then also distribution?
Yeah, I think I think by the end of this calendar year, these studies should be finished, and we'll be able to determine whether or not they were working or not. And if they do continue to have good results, and they're going to be produced, and it'll be several months for them to produce the vaccine. But I've heard that by early 2021, vaccine will be available. Now, Canada has pre-ordered from both of those, Pfizer and Moderna, has pre-ordered the vaccine, so that's good. But again, even once Canada gets the vaccine, who's going to get it first? Is it going to be people living in care homes? Is it going to be the frontline workers? I would suggest that young, healthy people, people that aren't getting very sick from SARS coronavirus infection, maybe they're not the highest priority, although they're the ones that are getting infected the most often. But it'll probably be to the spring, to the summer before we see the vaccine in most people's arms in Winnipeg, I would guess. And it's just a guess.
Okay, thank you. Another question came in, very interesting. It says, how does funding for COVID research compare to HIV research?
There's the Canadian Institutes for Health Research funds all research in Canada, and they came out with all of these calls for COVID research funding. And I think it's important to point out that they didn't take money away from any other research. They didn't take money away from cancer research or Alzheimer's research or HIV research. It was new money that the government put towards that. So that's good news. COVID money for COVID research is miles higher than any other infection or any other disease right now. Everyone is throwing everything at COVID that they can. I sit as chair for the advisory committee to the Canadian Institutes for Health Research, and there the CIHR gets $21 million a year for HIV research. And so that's a good amount, and it helps people like me do our work. But COVID research would be 10 to 100 times that for sure.
Because you're not able to travel to Africa right now, how is that impacting your ability to do your research, particularly with the cohorts of women that you've been talking about?
Yeah, one thing I'm really proud of is that over the years, we've built capacity at the University of Nairobi. So we have state-of-the-art labs there. And like I showed, we have Kenyan graduate students that are working there. And all of the clinics are run by Kenyans. So they don't need a Winnipegger to go over and run the projects for them. They can run them themselves. I'm involved and I give advice and things. But what really has hampered our research is when, like everywhere else in the world, they had these public health shutdowns and people couldn't, there was quarantines, and there was, people couldn't get out in the evenings and that really affected our research. Our clinics weren't open, and we're supposed to start our second phase of our aspirin study last spring, and that was canceled, and we're hoping to do it this year. So it's canceled our ability, it's affected our ability to do research, but more at a local level, not because I can't get over there, although I do go over a couple of times a year to help manage the projects.
And last question, we were talking a little before we started the session today, just about how much the COVID has impacted you and your fellow colleagues in the way of, the shift of your resources or your research time or whatnot. So, can you walk us through a little bit about how many people on your team are involved in COVID research within the Department of Community Health Sciences, Health Services?
Yeah, sure. So as I mentioned, we have 9 core faculty members. And I know that at least 7 of them, and maybe 8, are all shifted to do COVID research now in some capacity. They haven't, stopped doing their other research, but they've all shifted in some way or other to do COVID research. And our colleagues who we work very closely with at the National Microbiology Lab, for them, they've really focused completely on COVID. Some research has continued, but as Canada's premier virology lab, they're all hands on deck in terms of COVID. So a lot of people are throwing everything that they can at it. And those of us are doing the small things that we can. Like I said, I'm involved in a supportive role to a number of different projects, including one at the University of Florida, where we're getting samples and studying the antibodies and the immune system, and that hasn't started yet, so I didn't present it. But I think everyone is involved in it in one way or another in my department.
Super, thank you. Well, those are all the questions that we have. So I want to thank you, Dr. Fowke, for that really engaging and interesting presentation. Thank you so much. There's lots of great information in there. Thank you to everybody for participating. As I noted earlier, and as the usual process, we will make sure that this presentation is available to you very soon in a link. So you feel free to view it as many times as you like and share it with others. Again, we'll be sending you a link to a follow-up survey. Please do respond. We really do look at your feedback. It's the only way we're able to improve. So if you could join us next week when we will hear from Dr. Lara Rozanoff-Govin on the topic of, also COVID related, which is how to teach about a global pandemic during a global pandemic, fluxus and responsive teaching with anthropology now, COVID-19. This is a very new course that we launched in the fall here at the University of Manitoba. So there'll be lots of really great information on how that course is going and what she's teaching. So thank you again for your participation. Thank you for being involved and for spending the last hour with us. Stay safe, and we will see you next week. Thank you.